Okay, this will be something like a theoretical introduction to chlorophyll fluorescence in general. That can help you even with different other projects, maybe if you are working with remote sensing and these things. So if you take any kind of photosynthetic material, here is a leaf, and you illuminate it by white light or sunlight, then you see it's green. And this is a reflected light that is coming to your eye. Um, a lot of light that you don't see is absorbed by chlorophylls or other pigments. By chlorophyll, for higher plants analogy, the most absorption is in blue and red. That is why the arrow is something like bluish red. A part of the light is, of course, transmitted. I'm pretty sure that when you were a kid, you took some leaf and you were looking to the sun through that and you saw it reddish. So this is transmitted light. And the thing we don't see normally is a fluorescence. The fluorescence here is presented not as an arrow, it's not oriented. With this multi-arrow presentation, I wanted to tell you that the fluorescence is emanating to all spatial angles. So even though you illuminate your samples with a direct light or directed light from just one side, the fluorescence is going to all angles. What is the color of the fluorescence? Klaus Lichtenthaler, Klaus Buschmann, and Professor Lichtenthaler, they have a few very famous papers about plant autofluorescence, or they call it multicolor plant autofluorescence. And they have a quite nice graph, so I use it here. Even there exists much more other compounds now that are dedicated to plant autofluorescence. But in this graph, they use excitation light 355. It's a laser, UV laser. And they saw some blue fluorescence. Here it's marked as F440 because based on their spectral measurements, they selected few bands. And they realized they can measure a lot of features or a lot of reaction of plants based of combination of these bands. That the spectrum is not stable. It can change depending on plant status, age, treatment. So they saw some blue fluorescence that they called fluorescent 440. And this fluorescence is mostly from plant membranes, hydroxycinnamic acid. But later, another group of Zoran Cerovic, they also proved that you can even measure NADPH and NAD fluorescence from nicotine amides. And you can see also the fluorescence in plants that contains a lot of alkaloids. So there are different compounds behind the blue fluorescence. Another band in green, there is a little peak, or it's more often it's described as a green shoulder. It's approximately at the wavelengths 420. And this is for flavins, flavonoids, terpenoids. So these are the compounds that mostly are connected with some kind of secondary metabolism, like hydroxycinnamic acid from the cell walls, from secondary metabolism, and it's mostly reaction of plants to some kind of stress or UV treatment. And then we of course have red fluorescence, and this red fluorescence is a chlorophyll fluorescence. Again, you can measure two bands, or you can measure it more at 690, that is short wavelengths, that corresponds more to photosystem 2. Again, I will talk about that later. And you can also see the shoulder or the second peak more shifted to longer wavelengths at 730, 740. That belongs to photosystem 1. Here the peaks are very high. Normally they are not so high. Photosystem 1 is, or if you can see it, is less pronounced. And as I already mentioned, the shape of the spectrum, it can vary. So you can have a lower blue fluorescence and high chlorophyll fluorescence. This happens especially in young plants. They have chlorophyll, they are not stressed. Here on the image on the right side, you can see there are two photographs, black and white. And on the right photograph, where is red chlorophyll fluorescence, you can see the leaf in the left part of the image that you don't see in green region. Simply, there is a lot of chlorophyll. It's probably a healthy leaf, so you can see a lot of chlorophyll fluorescence, and no other fluorescences is so much visible. Then you can see the needles, and because it contains different terpenoids or so you can see the green fluorescence. All leaves upper, these are senescing leaves or maybe infected, I guess. And very typically, here you can see very typical image. On the right side, you can see the leaf in chlorophyll fluorescence, where you cannot see a part of the leaf. There is some lesion or something like that. And in the green or blue, you can see the high fluorescence in the blue-green region. The blue-green fluorescence and chlorophyll fluorescence and the ratios, they depend also on the status of the plants and the health status of the plants. The spectrum can also change if you measure fluorescence, for example, from up or down. It very much depends on how the light penetrates inside the tissue. That in the leaf, some leaves are very thick, some leaves are more are less thick. Your water plants and seagrasses, they probably do not have so many cell layers, like I show here, the palisade and the spongy parenchyma. And the short wavelengths of the light, UV and blue, they typically do not penetrate so inside. So you see the most fluorescence emanating from the most top layers. The left spectrum will have a big blue or green peak and lower red peaks if you measure it from the top layer. The red light can penetrate deeper, and the green light, because the green is not absorbed, it's mostly just reflected from the plants, it can even go to even deeper layers. So the ratios between the peaks, they can vary. And this is a very nice method if you find a paper from these guys or papers. And we also have this module where you measure a different spectral bands, blue, green, red, and infrared. And you get 4 images that you can combine in different ratios. This is a module that we also offer in our FluorCams as a multicolor module, plant module. Can give you some information about stress and plant status. In your system, there is only chlorophyll fluorescence, and you should be not sad because it's a very nice tool anyway. This is a very nice experiment, and you can try it in the lab. Normally, you don't see chlorophyll fluorescence emanating from the leaf. But maybe you have before tried to extract chlorophyll. And if you use the blue LED that you see on the picture and you illuminate your chlorophyll extract, you can see the reddish fluorescence inside. It's really visible by eye. As I said, chlorophyll extract of green plants and contain mostly just only chlorophyll A and B. So there are 2 Soret's band of absorption spectrum. This is very extraordinary in nature and in physics, that you have 2 bands for excitations. So chlorophyll absorbed in blue and red region. Here you can see for 30 approximately the peak, and second one is approximately at 660. And in the image on the right, in the image of the spectra, you might see also emission, fluorescence emission, that is longer wavelengths, but it's in red. So for this experiment, if you use red light, you will not see it. Your eye can hardly distinguish the red from a little bit deeper red. So emission is from 670, 80. Again, depends what you measure, if there is extract or it's a whole leaf. This is probably known for a lot of years. And even the chlorophyll fluorescence is known for a lot of years. Soon we will have 100-year anniversary. In 1931, two guys, Kautsky and Hirsch, they perform a nice experiment. Then they take the leaf that was adapted to dark, and they illuminate the leaf suddenly by the light, and they were looking at the scene through the dark red glass. And what they see, they saw chlorophyll fluorescence, but moreover, and hopefully it's visible. Sorry, yeah. This is a video, it should be visible. You can see the glow. You can see it's not steady state. What they found out was that chlorophyll fluorescence is not a steady state signal. It's something that is changing in time. It's a kinetics. Even they use constant light. Here in the image, you can see the red spot. There is a spot where was applied diuron, TCMU. So this was the first experiment which proved that the chlorophyll fluorescence is a kinetic signal. Then people started to think, okay, what is behind the kinetic? Because normally fluorescence, in most fluorescence fluorophore, it's just steady state. You illuminate your sample and you get some signal. It's not changing in time unless there is some chemical reaction behind. If you use more light, you get more signal. If you use less light, you get less signal. So there should be, for sure, some chemical reaction behind the chlorophyll fluorescence kinetics. Here is a simplified image of photosynthetic machinery. The kinetics originate in chemical reactions or biochemical reactions that are provided or done by photosynthetic machinery. You probably know from your studies that photosynthetic apparatus contains a lot of pigment protein complexes. Very simply said, there are two very core complexes. One is called PS2 on the left side, the lighter green, and one is called PS1. These complexes are very, very special in nature because they can separate, they are serially operated in a line, and they can perform primary energy conversion. It means if you illuminate these systems by light, so in pigment protein complex in PS2 and PS1, the light can be converted to chemical energy. The electron is liberated and it's moving over the photosynthetic machinery. So they separate charges. Both of these complexes, they have two parts. It's a core called reaction center. And what you see on the left better, there is some light harvesting complexes typically, denoted by LHC, light harvesting complex. And the role of these light harvesting complexes is just to gather light to help to deliver more light to reaction center. They work like funnels. So if the light is absorbed in one molecule, then the absorbed light is transferred to another chlorophyll molecule and another until it reaches reaction center. In the middle of the reaction center, there is an active chlorophyll. In photosystem 2, it's called it's called chlorophyll 680. In the reaction center PS1, it's called chlorophyll 700 for a longer wavelength absorption. And once the photon reaches reaction center, let's say reaction center in PS2, P680, then the electron is deliberated and transferred to the sequence of other photosynthetic protein pigment complexes to QA, QB, plastoquinone pool, complex B6F, plastocyanin to PS1, and so on. During this process, there is synthesized ATP, and the electrons that is missing inside the reaction center, it's filled from water splitting complex. So also the oxygen molecules are produced during the process. But what happens if there is too much light? Because this is a chemical process, so it should have some kinetic constants. If there is more light than needed for photosynthesis, this is called photosynthesis. So if there is more light than needed for photosynthesis, and electron transport chains is filled by electron, all the transporters are reduced, then the light is, then this energy that was absorbed is recombined and re-emitted, or plants somehow should get rid of that. One way to get rid of the excess energy is heat. So a part of the absorbed energy is dissipated as a heat, and part of that is emitted as a fluorescence. Of course, we live in a real world, so the fluorescence, the energy in a fluorescence has less energy than the original photons, so it's red shifted. The fluorescence is red. So this is the arrow on the left side. By the way, most of the fluorescence that you measure in native plants, it's originate from photosystem 2, most of that at room temperature. Here is some graphical presentation of what I already said now. This seems to be some kind of theoretical concept, but it will help you to manipulate with a sample later. So here we have some photosystem, either photosystem 2 or 1. If we illuminate, deliver some energy, light energy is absorbed. Then most of that is used for photochemistry or photosynthesis, carbon assimilation finally. Part of the energy is dissipated as heat. Let's call it non-radiative decay here. And a little part of the energy is emitted as fluorescence. There is only small portion of light that is emitted as fluorescence. At room temperature, typically this is just between 1 to 5% from the absorbed light. So really very little. That is why you cannot see it by your eye mostly, maybe even in dark, and there is a lot of fluorescence. Because it's mostly also lost in reflectance, in green reflectance. So if we now try to quantify it or write it in some kind of mathematical manner, if number 1 corresponds to absorbed energy, all of other processes should give you, if you just count them together, they should give you number 1. These are relative values, these are quantum yields. So the quantum yield of photosynthesis plus quantum yield of non-radiative decay plus quantum yield of fluorescence emission should give you 1. What happens if a photosynthesis is working well? That simply you have little heat dissipation and little fluorescence. And if the photosynthesis is not working well, then the light or energy that is absorbed should go some other ways, either heat dissipation or fluorescence. So typically, if you put your samples under the FluorCam and you have very nice fluorescence, it means something is wrong with the sample. Don't be happy. And this inverse relationship, it allows you to, based on the measurement of fluorescence, it allows you to get some information about photosynthesis. That is very practical. So again, when photosynthesis in plants is highly efficient, the fluorescence is low. And conversely, if plant capacity in photosynthesis is compromised or saturated, for example, during a saturating pulse application, the fluorescence is high. Again, the concept from the other view. Here is a Kautsky effect, which I already mentioned. What they saw, they saw the kinetic behavior of the fluorescence in a dark adapted sample illuminated suddenly by a light, constant light. They saw that the fluorescence increase in the beginning. It reaches something like a maximum, and then in decline, and it declined in span of approximately 1 to 2 minutes to some kind of steady state. And to honor these guys, this effect is very typically in this field called Kautsky effect. What happens there? During the initial increase, fluorescence rises simply because photosynthetic apparatus is not working and the Calvin cycle is not working. It takes some time. And this increase is typically quite fast. It's in the range of seconds, 1 second, 2 seconds to reach the peak value. And the QA at the moment is, so if the Calvin cycle is not working, so electron from electron transport chains, they are not moving forward. All of the transporters are in reduced state. The photosynthetic apparatus cannot take more light, cannot convert more light to energy, to chemical energy. So the light is dissipated as fluorescence in this case. And fluorescence is increasing in the beginning. After some time, relatively short time, the Calvin cycle is activated, started, and it drains electron from the electron transport chain. So that is why in literature you can find a term photochemical quenching. It's in the terms that you quench fluorescence by photochemical reaction, by photosynthesis. And because the photosynthesis start to work more, the fluorescence is declining. Or of course, heat dissipation is another process. Yeah, so as I said, if you measure fluorescence, you are getting information about photosynthesis. Of course, you can distinguish between three processes, because it's interplay of three processes: fluorescence, photosynthesis, heat dissipation, and fluorescence. And there exist techniques and protocols that help you to distinguish between these processes. Here is another very nice experiment. This is a fast chlorophyll transient, and even you probably will never measure it. I will show it because it's a very nice animation. On the left side, we have a graphical presentation of the measurement. This is a measurement that is done with single turnover flash. Single turnover flash is something that is very, very strong. Strong pulse of light, and is very short, typically just up to 50 microseconds. But it brings a lot of photons. Normally, if you measure light, you measure something like 100 micromoles. But in this pulse, you concentrate 100,000 micromoles. It's really a strong pulse. And the reason for such strong pulse is for a very short time to saturate all reaction centers, all PS2s in photosynthetic materials that you measure. So this is a black line on the left side in the graph. What you see, it's a single turnover flash. And on the right side, on the right image, you can see this short and very fast experiment refers and can be nicely modeled by the processes in the photosystem 2. That is why on the right side, you can see just the photosystem 2, this reaction center, active chlorophyll P680. Here is also nice representation of the antenna system around the reaction center of photosystem 2. And you can see oxygen evolving complex here and plastoquinone transporters, QA and QB only in a photosynthetic membrane. So what happens? In the beginning, let's say we have a dark adapted sample. And with a very weak pulses, called measuring pulses, we measure something like a basic fluorescence, lower level of fluorescence, or it's very often called F0 fluorescence. These are short and weak pulses. When we apply the strong pulse, so you could see that photosynthetic machinery is working. Measuring pulses is a light that from definition should be enough weak that no reduction of photosystem of QA and QB transporter occurs, or there is just a basic amount of reduction and not statistically in some photos some complexes. And the electron, so always you illuminate with this amount of light, you can measure something and you don't see any kinetics. If you apply the strong light, what happens? All reaction centers, they are simply reduced. All QA is simply reduced. So at this point, even if you get more light, you cannot do more photosynthesis. Photosynthetic machinery is simply oversaturated and all transporters are reduced. This is a special state of the photosynthetic apparatus. And what happens? The energy is dissipated as a fluorescence, and you can measure maximum fluorescence at this point. This is so short experiment in terms of microseconds that you do not activate heat dissipation. Later, of course, Kelvin's, this is a dark phase again. After a strong light, you measure with a fast detector and you see how the fluorescence is declining until it reaches some steady state. First of all, the steady state is a little bit higher than originally dark adapted value of F0 because simply this decline is done because simply the light is used in photosynthetic machinery. And if the light is used, then you can see the decline in fluorescence. Advantages of chlorophyll fluorescence. First of all, it's to measure chlorophyll fluorescence. Fluorescence is optical signal. Optical measurements is always non-invasive methods. So you just can monitor some samples without touching it, without destroying it. So this is a first and big advantage of chlorophyll fluorescence. Even chlorophyll fluorescence is indirect. It can provide valuable information about photosynthesis. And photosynthesis is something like a fundamental process in plants and in photosynthetic materials. So it gives you information about the most important process in plants. This process, photosynthesis, is like our heart beating, is the most important process, and it's very sensitively reacts to different treatments, physiological status. It's very nicely and rapidly responds to growth condition. It depends on developmental stage. It can give you information about recovery from the stress, rapid information about the stress. So if you are working with plants and you are studying some stress responses or reactions to some kind of chemicals, of course you can see it later by eye that your plants are getting yellow or they have not good turgor or something like that. But if you are able to monitor photosynthesis, you have valuable information that is very rapid. And this is the information provided by chlorophyll fluorescence. Of course, PS2 activity is very sensitive to different biotic and abiotic factors. And the most important thing, it's very rapid. If you even press the plants by your hand, by your fingers, you can immediately see it in chlorophyll fluorescence transient, and for sure you will not see anything by your eye, or maybe even later you will not see the changes in the pigments. Okay, by chlorophyll fluorescence kinetic, you can detect stress responses very early, much earlier than by other methods. For example, hyperspec is another optical method, but the hyperspec gives you information about the colors. So in other terms, it gives you information about pigments. And to change the compounds and the pigments inside the tissue, it takes hours, even days. But chlorophyll fluorescence gives you information about photosynthesis, and it's rapidly responding to anything. So very popular applications of chlorophyll fluorescence nowadays, screening of mutants, and analysis of different genetic variations from the naturally occurred variants in nature. Of course, studying ecological diversity, it's a very good indicator of the stress. Detection of biotic stress, infection, and nowadays also a lot of people use it to improve crops and other plants, because photosynthesis is a core process that gives you, if it's working better, or if you can keep it in the state it's working on the maximum level, of course you can produce more, you can fix more carbon, and you can have larger plants.